Rh antigen is found on the wall of the RBC, then it is called Rh positive. When Rh antigen is not found on the wall of the RBC, then it is called Rh negative. Now regarding the specificity of this Rh positive and Rh negative grouping, the most important thing is Rh group is controlled by a allele called D. And this antibody is called entity. Entity. So the gene corresponding to antigen D, gene corresponding to antigen D is called that means allele is also D. And remember this thing, if D is absent, its place is occupied by an alternate form called small d. Is it clear, please? Rh antigen is found on the wall of the RBC. It is called D. And its antibody is called NTD. When Rh antigen is not found, it is replaced by an alternative form of the allele. If D gene is absent, its place is occupied by alternative form. Alternative form means an allele called small d. That means Rh positive individual may be found in this form or in this form. Both are under allele. Both are either capital D or capital D small d type. When the allele form is Rh negative, it is capital D, So if you remember, this D allele is dominant over small d. D allele is dominant over small d. Is it clear to everybody? Is it clear? Is it clear now? Rh grouping? Rh grouping based on the presence of Rh antigen on the wall of the RBC. If Rh antigen is present on the wall of the RBC, then the blood group is said to be Rh positive. If Rh antigen is not found to be present on the wall of the RBC, the blood group is said to be Rh negative. 85% of the human population are found to be Rh positive, and only 15% of the human population are found to be Rh negative. So here you see one thing, Rh positive or Rh negative, whatever it may be, the main thing here is it is controlled by a specific allelic form, its inheritance. The allele is called the D. So the gene controlling the Rh positive Rh blood group controlled by gene called D. D may be found in two alternate forms, capital D and small d. That means capital D is found to be dominant over the small d. That's why when Rh positive individual should have the genotype either capital D or capital D small d. Whereas Rh negative individual should have the genotype small d. Is it clear?
Yes, sir, clear.
Okay. So it's clear to everybody. Rh positive and Rh negative. Now, regarding the inheritance of this. So I am to rub it out now. Complete it please quickly. Capital D is homozygous. Capital D small d heterozygous. Both are Rh positive. Happen if father is what will happen to the child if father is Rh positive and mother is Rh negative? In other words, we can say, now suppose father Rh positive, Rh negative. So this is the first child. If Rh positive, because if father is Rh positive and mother is Rh negative, it is not always true that the first child will always be Rh positive, because you know father genotype may be like this. Look, Rh positive and Rh negative. So if father is Rh positive, then mother, suppose Rh negative, then also every child may not be Rh positive. So 50% of their child are Rh positive and 50% are Rh negative. But if father is homozygous Rh positive like this, and mother is Rh negative, then 100% will be Rh positive. We don't know whether father is Rh positive or Rh negative. That's why we cannot say directly that every child would be Rh positive positively. So, what should we write? First child if Rh positive. Then what happens? Now you know, normally mother's blood cannot be mixed with the fetal blood during pregnancy due to the presence of placental barrier. Listen carefully. Due to the presence of placental barrier, mother's blood cannot be mixed with the fetal blood. Due to the placental barrier, mother's blood cannot be mixed with fetal blood. First point. Okay. If it is so, then when the mixing is possible, only at the time of parturition, that is childbirth. Only at the time of parturition, that is childbirth, mother's, what is found? Only at the time of parturition or childbirth, mother's blood comes in contact with the fetal blood. How? Because at the time of parturition, at one time, the amniotic membrane covering the fetus gets ruptured. The amniotic membrane covering the fetus gets ruptured. And as a consequence of which, what happens as amniotic membrane gets ruptured, the mother's blood comes in contact with the fetal blood. What happens? Mother's blood comes in contact with the fetal blood. And as a consequence of which, in the mother's blood, NTRH factor or NTD started to be produced. So,
सर ये पॉइंट की लिखते लगबे
wait. During parturition. During parturition, mother's blood comes in contact with the fetal blood. So what happens? NTRH factor NTRH factor is started to be produced in the mother's body. The mother's immune system start producing NTRH factor. Mother's immune system starts producing NTRH factor. So mother's immune system starts producing NTRH factor. But you know, but in the meantime, the process of parturition is completed. Till NTRH factor is developed in the mother's blood, the parturition process is completed, so the first cell is found normal. First cell is found normal because you know only mixing occurs at the time of parturition. So till NTRH factor is developed in the mother's blood, parturition process is completed. Hence, first child is born normal. Clear? Is it clear to everybody?
सर ड्यूरिंग parturition ऐसा का टटू बोलना पड़
Look one thing, at the time of parturition means childbirth. So whenever mother's pituitary starts secreting oxytocin, vigorous contraction of the uterine wall takes place. And at one time, the membrane covering the fetus called amniotic membrane gets ruptured. As soon as amniotic membrane gets ruptured, mother's blood comes in contact with the fetal blood. Is it clear up to this?
Yes, sir.
Once mother's blood, so when this mixing occurs, just at the time of parturition, when parturition already started. So mother's immune system then starts producing NTRH factor in his body, but in the meantime, the process of parturition completed, right? Someone is calling.
वेटिंग रूम में क्या जाने आज
As soon as mother's blood comes in contact with the fetal blood, anti-Rh factor starts destroying anti-Rh factor starts destroying fetal RBC. Destroying fetal RBC. As soon as mother's blood comes in contact, বলো
as soon as এর পরে কি লিখলে
mother's blood comes in contact with fetal blood, anti-Rh factor starts destroying fetal RBC. Anti-Rh factor starts destroying fetal RBC. Clear? As soon as mother's blood comes in contact with fetal blood, anti-Rh factor starts destroying fetal RBC. Clear?
Yes, sir.
Leads to erythroblastosis fetalis. Leads to erythroblastosis fetalis. You clear? And this anti-D or anti-Rh factor is found to be present up to 1 week after birth and starts destroying fetal RBC continuously. Are you getting me? When mother's blood comes in contact with the fetal blood, in this case, then anti-Rh factor already developed in the mother's blood starts destroying fetal RBC, leading to erythroblastosis fetalis. This anti-D or anti-Rh factor is found to be present in infant's blood for at least 1 week after birth and starts destroying more and more RBC. Actually, the child becomes Rh positive, the same thing will happen. Clear to everybody? Is it clear?
yes sir
So this is what, now what to do? In order to check this, if father is found Rh positive and mother is found Rh negative, then after the birth of the first child, and if the first child is found Rh positive, then mother is given an Rh immune globulin injection to prevent the formation of antibodies in the mother's blood. Prevents active antibody formation by the mother. Is it clear? If how to control this? If father is Rh positive and mother is Rh negative and first child is found to be Rh positive, then after the birth of the first child, the mother, after the birth of the first child, blood is his or her blood group would be tested, and if it is found Rh positive, then mother is given one immunoglobulin, Rh immunoglobulin injection to prevent the active antibody formation by mother. Is it clear to you? Is it clear? How to control it? After the birth of the first child, if the first child is found to be Rh positive, then mother is given Rh immunoglobulin injection to prevent active antibody formation by mother. Clear?
Yes, sir.
Okay. So it is completed. Now we have to proceed towards the blood coagulation. Blood coagulation. Formation of prothrombin activator. Conversion of prothrombin to thrombin. Formation of fibrin. There are three main steps of blood coagulation. Formation of prothrombin activator, conversion to of prothrombin to thrombin, and formation of fibrin. For this, to carry out blood coagulation, a number of blood coagulation factors are to participate. So you need to remember them also. Blood coagulation.
একজন মনে হয় ওয়েটিং রুমে আছে।
Factor 1, fibrinogen. Factor 2, prothrombin. Factor 3, thromboplastin. Factor 4, calcium ion. Factor 5, labile factor. Factor 6, convertin. It is of no use now. It is of no use.
5 নম্বরটা কি লিখেছো?
Labile factor. Proconvertin.
Sir, 6 numberটা কি?
6 number convertin, but it is of no use now. It is of no use. A kind of hypothetical factor. Okay. Plasma thromboplastin antecedent Factor 13. So they all are to take part. Huh? Really taking part in the process of blood coagulation. Clear? Now I have to show how do they take part. Can I rub it out? এই কামড়টা মতো দিব। Blood coagulation formation of prothrombin activator. It is produced by two pathways. Can I rub these coagulation factors, please? Antecedent, 12 Hagman factor, and 13 fibrin stabilizing factor.
হ্যাঁ স্যার হবে, এবার রাফ করে ফেলি।
Extrinsic pathway. This pathway is triggered by injury to vessel wall. What happens here? Injured tissue factor 7A, factor 3, factor 10A. Ten A plus factor 5A, prothrombin activator. Is it clear? This is the extrinsic pathway. And what happens in the intrinsic pathway? Six, where is 6? 5. This one. 7 are pro-converting. A means active factor 7. The term A is used to indicate that it is active factor 7. Now, is intrinsic pathway. Intrinsic pathway, it is stimulated by it is triggered when blood is exposed to collagen fibers. Now you see what happens. Collagen fibers of ruptured endothelium of blood vessel. Surface contact.
স্যার অবৈধ ওয়েটিং রুমে আছে।
Factor 11, then factor 11, active factor 11. Factor 9. Factor 10 একটা বেদা একটু আসছে আমি Is it clear? Look at this.
সেটা কি লিখছো প্লেটলেটের পরে?
Huh? Platelet phospholipid. Fibers of ruptured endothelium of blood vessel, whenever comes in surface contact with factor 12, the factor 12 would become active factor 12. That in the presence of factor 12, factor 11 is converted to active factor 11. Then this active factor 11 stimulates factor 9 to become active factor 9, which is called Christmas factor. Now, factor 10 in the presence of active factor 9, active factor 8, and platelet phospholipid convert to active factor 10. Now, active factor 5 and calcium ion stimulate the conversion of active factor 10 to prothrombin activator. Any doubt? You can tell me. Please. is actually stimulated by the factor 9 itself. So the first step is completed. This is the formation of prothrombin activator. Now I'm going to rub it out, please. Conversion of prothrombin to thrombin. What happens? You know, prothrombin is produced in liver with the help of vitamin K. Vitamin K helps in the synthesis of prothrombin in liver. Hence, prothrombin is called. Hence, you know, in this way, vitamin K helps in blood coagulation. Remember this thing. Vitamin K helps in the synthesis of prothrombin in the liver. And in this way, vitamin K helps in blood coagulation. Because prothrombin in turn is converted to thrombin, the active proteolytic enzyme. Prothrombin is converted to thrombin, which is an active proteolytic enzyme. And here, prothrombin activator. Then formation of fibrin. Fibrinogen protein in the presence of thrombin convert to fibrin monomer plus fibrinopeptides. Fibrin monomer is found to become soluble. So now in the meantime, factor 13 in the presence of calcium compared to factor 13A. Next, monomer (13A) form fibrin clot. Form fibrin clot. Then within 5 to 30 minutes of fibrin clot retraction occurs. In clot retraction, 40% of the volume decreases and coming out as serum, a pale yellowish fluid. Once serum comes out, this is the end of blood coagulation. দেখো বুঝতে পারছি কিনা? Sorry. fibrinogen in the presence of thrombin, fibrin monomer plus fibrinopeptides. Then factor 13 in the presence of calcium ion would become active factor 13. In the presence of active factor 13, fibrin monomer will be forming the fibrin clot. There is now it is insoluble. Then within 5 to 30 minutes of the clot formation, clot retraction occurs so that 40% of the volume of the fibrin clot decreases and coming out in the form of serum. And then you can say that blood coagulation process is completed. Is it clear? Or even a doubt? Clot retraction, serum. So change it and I like this, it's better. Clear?
Yes, sir.
Yes, sir.
Then the next one is fibrinolysis. The process of the process of dissolution of fibrin clot is called fibrinolysis. The process of dissolution of fibrin clot is called fibrinolysis. And how does it occur? Plasmin or fibrinolysin form fibrin degradation substance. Just comes to the after that what will happen? Now this fibrin degradation substance in turn inhibit thrombin. The fibrin degradation substance in turn inhibit thrombin, and so that thrombin can't able to coagulate or degrade further fibrinogen. So this is also a kind of feedback. Fibrinolysis is a process of dissolution of fibrin clot, and it is carried out in the presence of the enzyme fibrinolysin, and fibrin clot becomes fibrin degradation substance, and this substance in turn actually inhibit thrombin to maintain homeostasis. Now you see here plasmin, from where this plasmin comes? Plasminogen is converted to plasmin in the presence of tissue plasminogen activators. TPM is or fibrinolysin. Clear? Produced from the plasminogen in the presence of tissue plasminogen activator. Plasminogen is converted to plasmin and causes the dissolution of fibrin clot, forming fibrin degradation substance, which in turn inhibit thrombin. Because thrombin is also required to, once blood coagulation process is completed, the thrombin produced from prothrombin is also required to be inhibited. Otherwise, it will further degrade fibrinogen. That's why, as a feedback mechanism, thrombin is required to be inhibited, and it is inhibited by fibrin degradation substance produced as a result of dissolution of fibrin clot. Clear?
Yes, sir.
Okay, that's fine.
Sir, TPR full from
Tissue plasminogen activator, written here. Tissue plasminogen activator. Basically, thrombin can also stimulate it. TPA or sometimes even thrombin. That means plasminogen can be converted to plasmin either in the presence of TPA and thrombin both. Where tissue gets damaged, TPA is released. From where TPA will come? TPA will come from damaged tissue. Damaged tissue produces TPA. TPA causes the conversion of plasminogen to plasmin, or even thrombin can also help in the conversion of plasminogen to plasmin, so that ultimately dissolution of fibrin clot occurs. So once, what the benefit of it? Once fibrin clot is required to be dissolved, and secondly, the product of this dissolution is required to inhibit the thrombin. So this is what happens in blood coagulation. Now you can say the process of blood coagulation is completed. Clear?
Yes, sir.
Muscle. Thank you very much. Name is called modified tissue fluid. First of all, we need to know how is lymph formed. Look one thing. Excellent. See the tissue capillaries. When blood is flowing through the tissue capillaries, then the plasma part oozes outside. 90% of which will return to it again, and rest 10% will form the lymph. I am repeating it again. While blood is flowing through the tissue capillaries, as the tissue capillaries are very thin wall, the plasma part oozes outside, although 90% of which will return to the blood again, but rest 10% gets modified to form lymph. lymph will then be carried through lymphatic duct and eventually enters the venous system, the vein. Lymphatic vessel. And in the lymphatic vessel, you will get one-way valve. So that once lymph enters here, cannot be returned to the earlier place. So this is how lymph, well, I'm telling you again, when blood is passing through the capillaries, then what happens? A part of this will be converted to a modified tissue fluid called lymph. Lymph is carried by lymphatic vessel, and in the lymphatic vessel, you will get some valves, and these allow one-way by ensure one-way flow of lymph. This is what a lymph, and this is a modified tissue fluid. It actually acts as a accessory medium of test when the areas of our body. Basically, remember this, there are some areas in our body where blood can't reach. The areas in our body where blood can't reach, the lymph reaches and carry oxygen and nutrients to this area, and also carry carbon dioxide and excreted products from that area. So this is what the basic role of the lymph. Are you getting me? Is it clear how is lymph formed? How
Yes,
is
sir.
lymph formed? Or in doubt? Is it clear?
Yes, sir.
So remember this thing, lymph is to play certain most important role. Now, composition of lymph. What it is made of? Lymph plasma, and lymph corpuscles. And lymph vessels, no, through the capillaries again. It enters the blood, enters the vein, not directly joined to the vein, through capillaries. I am showing here, but remember this thing, through capillaries it is to enter the vein again. Are you getting me? So lymph vessels, lymph plasma, actually the amount of calcium is less. You see, in comparison to blood plasma, the amount of calcium is less. Lymph plasma, then blood plasma. Remember this thing. Next, many globulin proteins are present as antibody, and lymph corpuscles includes the leukocytes only, mostly lymphocytes. Lymph corpuscles include lymphocytes. Are mainly lymphocytes. Limb corpuscles, what are these limb corpuscles? Limb corpuscles are mainly lymphocytes, remember these things. Erythrocytes are not present. And platelets, limb. Erythrocytes and platelets are not found in limb. Limb corpuscles are the main component. Limb corpuscles, mainly the lymphocytes are found. And so, why is function of limb? Limb carries nutrients and oxygen in those areas of the body where blood cannot reach. Limb is to carry oxygen and nutrients in those parts of the body where blood cannot reach. And secondly, limb is actually, it is also to carry excretory production carbon dioxide from those parts of the body also. It produces lymphocytes. So it got a number of antibodies. So it destroys different invading microorganisms. As it is lymphocytes, so it can destroy different invading microorganisms. The main function, it acts as accessory medium of transport. Why it is called accessory medium of transport? Those areas in our body where blood cannot reach, the limb reaches and helps in what? Helps in carrying oxygen and nutrients to those areas, and also to carry carbon dioxide and excretory products from that area also. Secondly, as you know, as they possess lymphocytes, they can destroy invading microorganisms. Limbs helps in maintaining volume of the blood also. Limbs helps in maintaining volume of the blood. Clear?
Yes, sir.
Okay. So this is what about the limbs. And largest limb gland. Spleen. Now regarding the pigments, and it is very simple, you can go through it very simply there, so that in the next class I can start the muscle. Okay?
Sir, একটু সাইড
Oh.
ডান স্যার
Okay. So function, what I say? For the functions, accessory medium of transport helps in destroying invading microorganism, has to maintain the volume of the blood. So this is all for today. In my next class, and then
স্যার হেল্পস ইন ইনভেডিং কি বলেছিলে
কি?
স্যার লিম্পের ফাংশনটা হেল্পস ইন ইনভেডিং
invading microorganisms.
আচ্ছা স্যার
Destroy invading microorganism and foreign particles. And lastly, helps in maintaining the volume of blood. Clear?
হ্যাঁ স্যার
Okay. So that's all for today. In my next class.